Okay, hello everyone. So here we are continuing with our story on metabolism of amino acids. So if you remember, so far we have completed the metabolism of nitrogen from amino acid. Last time we discussed about the metabolism of carbon skeleton of amino acids, and now we're going to discuss about the biosynthesis of some compounds which precursors are actually amino acids. So for this specific video I have chosen to tell you about the biosynthesis of heme, glutathione, creatine and serotonin. But let me tell you at the beginning, there are much more. There are more molecules which are synthesized from amino acids, but probably we're going to discuss, not probably, but for sure, we're going to discuss about their biosynthesis and biological function in some of the upcoming chapters. But let's start with the biosynthesis of heme. You know very well that heme is the prostatic group of a large group of heteroproteins which are called heme proteins. And some of the famous molecules belonging to this group are hemoglobin, which is the major transporter of gases through organism and involved as very important physiological buffer in the maintenance of acid-base equilibrium. Then we have myoglobin with kind of similar function to hemoglobin. Then we have cytochromes, especially cytochrome P450, which is involved in biotransformations of numerous exogenous and endogenous substances and metabolites. Then we have certain enzymes containing heme as a prostatic group like catalase, and so on and so on. The structure of heme is very well known probably to you from the part of the general biochemistry. So it contains a protoporphyrin ring, which actually contains 4 pyrrole rings, which are connected by methynyl bridges. If you take a look here at the structure of one porphyrin, you see that those double bonds are conjugated, meaning separated by one single bond, which stabilize the structure of porphyrin, plus provides a very specific red color of this pigment. In the central part of porphyrin ring, 2 valent iron ions are incorporated, forming, as you know, 6 bonds, 4 to nitrogens from pyrrole rings, 1 to histidine from globin chain in hemoglobin, and 1 for oxygen. But we are focusing on porphyrin ring and heme itself. So if you can see, hydrogen atoms are actually substituted with certain atomic groups. And those atomic groups are methyl groups. So here we have 1, 2, 3, 4 methyl groups in this structure. Then we have acetyl group. This one is not shown here because it's converted to methyl group during biosynthesis. Then we have propionyl group, and here we have 2 propionyl groups, and vinyl groups, and here we have 2 vinyl groups which are derived from by decarboxylation of propionyl group during synthesis. So very specific arrangement of those atomic groups attached to pyrrole rings. Because of those different kind of groups attached, or hydrogen atoms substituted with different groups, there are different porphyrin types available, and each of those types has different isomers. So we can talk about uroporphyrins, coproporphyrins, and protoporphyrins. So uroporphyrins, they contain 4 acetyl and 4 propionyl groups. So if you calculate all of these, it gives us 8 carboxyl groups in the structure. And this type of porphyrin is the most soluble one, and it's excreted predominantly in urine. Coproporphyrins are slightly different because of 4 acetyl group. Now we have 4 methyl groups, so the number of carboxyl groups is decreased to 4 carboxyl groups, and this molecule is more lipophilic. And finally, protoporphyrins, they have 4 methyl groups, and 2 propionyl groups are decarboxylated to 2 vinyl groups, and then we have 2 propionyl groups remaining. So Each of these porphyrins actually has 4 potential isomers, but only one, usually isomer number 3, is physiologically important. So protoporphyrin 3 is actually protoporphyrin 9 according to the old nomenclature, but we are still using it. So we can conclude the very well-known definition of heme, that heme is protoporphyrin 9 with iron ions incorporated in the central part of the structure. Reduced form of those porphyrins are marked as porphyrinogens, and those are molecules which are intermediates of biosynthesis of porphyrins. So in these molecules, instead of methanyl bridges, we have methylene bridges, and those protoporphyrinogens are colorless molecules and they're unstable molecules. So free porphyrins do not have any biological functions, but they are interesting for from a laboratory point of view, for laboratory determinations, because the quantification of those types of porphyrins is valuable for diagnosis and the description of different disorders marked as porphyrinopathies like porphyrias or porphynuриas. But at this very moment, I'm not going to go into details about these disorders and type of those porphyrinopathies, because you're going to do that later on or throughout some other subjects. So the biosynthesis of heme occurs in all cells generally, but the most frequent biosynthesis occur in bone marrow, which is logic because of the biosynthesis of erythrocytes and hemoglobin. So the whole part and pathway occurs partially in mitochondria and partially in cytosol, and actually this pathway begins in mitochondria and ends in mitochondria. It consists of the total number of 8 enzymatic reactions, out of which 4 reactions are found in mitochondria and 4 in cytosol. Speaking of the origin of carbon and nitrogen atoms, all atoms of protoporphyrin ring originate from a non-essential amino acid, glycine, and succinyl coenzyme A, which is an intermediate of the Krebs cycle. And actually the first reaction of the biosynthesis of heme is the condensation of those 2 molecules taking place in mitochondria. So glycine reacts with succinyl coenzyme A to form delta aminolevulinate, or ALA, or a more correct option would be 5-aminolevulinate, but still this common way of marking like delta is more famous than 5. So the enzyme catalyzing this reaction of condensation is delta aminolevulinate synthase, or ALA synthase, and it requires pyridoxal phosphate coenzyme for its action. Actually, this first reaction occurs in 2 phases, in 2 steps. So in the step number 1, glycine reacts with succinyl coenzyme A to form one intermediate alpha-amino beta ketoadipate, and this first phase requires PLP coenzyme. In the step number 2, alpha-amino beta ketoadipate is decarboxylated to delta-aminolevulinate. This reaction is regulatory reaction. So ALA synthase is actually major regulatory enzyme of the metabolic pathway. And this one is inhibited by feedback inhibition. So the products like heme are actually inhibiting the action of ALA synthase. And 2 mechanisms are observed. One is allosteric feedback inhibition, and the other one is impact on transcription and translation and the biosynthesis of the enzyme itself. So if we take a look at the overall metabolic pathway, here we are, and this one represents each pathway towards the biosynthesis of heme. So here we are at the very beginning. Reaction number 1 occurs in mitochondria. So succinyl coenzyme A and glycine condensate to form delta aminolevulinate, and 1 carbon dioxide is released from the system. So let's explain in short reaction number 2, and then we will see a little bit of details in the next slide. So in the reaction number 2, first we have to transport ALA from mitochondria to cytosol. So reaction number 2 actually takes place and occurs in cytosol. And this reaction means the condensation of 2 ALA molecules. So take into account that we're going to have 2 carbon dioxide here. So 2 delta ALA condensate to form porphobilinogen. And the reaction is catalyzed by ALA dehydratase or porphobilinogen synthetase. This enzyme also requires some SH group and some zinc ions. So let's get a little bit an overview of details about this reaction. So synthesis of porphobilinogen, PBG. So delta aminolevulinate is transported from mitochondria to cytosol, and it undergoes condensation to form porphobilinogen. And here, because this condensation is asymmetric, we're going to have, as you can see, we're going to have different groups attached, and those are propionyl and acetyl groups attached to porphobilinogen. The enzyme is delta aminolevulinate dehydratase, ALA dehydratase, or porphobilinogen synthase. Both synonyms are equally used. So this enzyme requires the presence of SH group and zinc ions, and because of this, this enzyme ALA dehydratase is especially vulnerable and sensitive to heavy metals poisoning, especially to lead poisoning, because lead actually inhibits the activity of this enzyme, and as a result there is an accumulation of delta aminolevulinate and its consequent appearance in urine. Apart from anemia, which is the major symptom of lead intoxication, also severe psychiatric symptoms are observed because delta aminolevulinate structure is very similar to the structure of one neurotransmitter, and that is GABA, gamma-aminobutyric acid. In the reaction number 3, let's get back to our story on the synthesis. So porphobilinogen undergoes condensation by means that 4 porphobilinogens condensate to form uroporphyrinogen. And if you can see, this reaction can go in 2 steps and in 2 directions. So in reaction number 1, there is uroporphyrinogen synthase, which yields the formation of uroporphyrinogen structure, which is an open chain of tetrapyrrole, and then under the spontaneous cyclization by synthetase alone, we get uroporphyrinogen 1, which is not physiologically active. But if we have the second step catalyzed by co-synthase, we're going to have physiologically active uroporphyrinogen 3, which will continue the synthesis towards heme. Notice that in uroporphyrinogen 3, acetyl and propionyl group at the 4th pyrrole ring changed their positions. So let's summarize reaction number 3 occurring in cytosol. So reaction number 3, synthesis of porphyrin ring occurs in cytosol. So 4 porphobilinogens tend to condensate to form uroporphyrinogen 3 by the enzyme uroporphyrinogen synthase or porphobilinogen deaminase. So that is actually the condensation of 4 BPGs, followed by deamination, thus forming linear molecule of tetrapyrrole. In the second phase, you can have two options. Spontaneous cyclization, which is completed non-enzymatically, yields the formation of uroporphyrinogen 1 without any physiological function. But if this tetrapyrrole undergoes enzymatic cyclization by the enzyme uroporphyrinogen 3 cosynthase, you have the exchange of acetyl and propionyl group at the fourth pyrrole ring, yielding the synthesis of uroporphyrinogen 3, which continues the synthesis towards heme. Let's move on. So here we are at uroporphyrinogen 3. So now we are still in cytosol, and uroporphyrinogen 3 is converted to coproporphyrinogen 3 by uroporphyrinogen decarboxylase. So 4 carbon dioxide molecules are removed from propionyl groups to form vinyl groups. So in total, 6 carbon dioxide molecules are going to be removed during the biosynthesis of one molecule of heme. And let's summarize it. So the reaction number 4 is synthesis of coproporphyrinogen, again taking place in cytosol, and uroporphyrinogen 3 is converted to coproporphyrinogen by uroporphyrinogen decarboxylase. Acetyl groups are decarboxylated to methyl groups. Coproporphyrinogen 3 is then transported to mitochondria, so the rest of the reactions take place in mitochondria. So here we are. Now coproporphyrinogen 3 is converted to protoporphyrinogen 9 by coproporphyrinogen oxidase, and this reaction occurs in mitochondria where 2 propionyl groups are converted to 2 vinyl groups. And here is the summary. So reaction number 5, synthesis of protoporphyrinogen 9, occurs in mitochondria, and reaction is catalyzed by enzyme coproporphyrinogen oxidase, and 2 propionyl groups are decarboxylated to 2 vinyl groups, and the total of 6 carbon dioxides are released from the system. Let's move on. We are still in mitochondria, and protoporphyrinogen 9 is converted to protoporphyrin by protoporphyrinogen oxidase. And if you can see now, we are changing those methylene bridges to methanol bridges, which are characteristic to protoporphyrin structures. Also, now we see that those double bonds are conjugated, separated by one single bond, which actually contributes to a higher stability of a molecule. So let's summarize. Reaction number 6 is synthesis of protoporphyrin 9 occurs in mitochondria by the catalytic function of protoporphyrinogen oxidase. Methylene bridges are oxidized to methanol bridges, and it results in conjugated double bonds, which are giving more stable molecule. And the last reaction is incorporation of iron ions, 2-valent iron, in the molecule of protoporphyrin to give the functional molecule of heme, and the reaction is catalyzed by ferrochelatase, and this is the last reaction occurring in mitochondria. So this enzyme is located in inner mitochondrial membrane, and it requires the presence of certain reducing agents like ascorbic acid and cysteine. It's also inhibited by lead, but this time the inhibition is indirect through prevention of reduction of ferri to ferroions. Biosynthesis of heme is regulated in different ways depending on the tissue in which the regulation occurs. So in liver, regulation is focused on activation and inhibition of ALA synthase, which is the major regulatory enzyme, by the terms of feedback inhibition, which can be accomplished by either allosteric or enzyme synthesis regulation. This enzyme synthesis is stimulated also by certain medicines like barbiturates and narcotics, because they are extensively biotransformed by cytochrome P450s in liver, which contain heme. So there is an excessive usage of heme, which actually leads to the excessive and enhanced biosynthesis of ALA synthase. Also, the synthesis of this enzyme is stimulated by certain steroid hormones like testosterone and oral contraceptives. The second way to regulate heme biosynthesis occur in erythroblasts, because those cells can accumulate large amounts of heme. So it's pointless to have heme as a molecule for feedback inhibition at this very point. So actually other enzymes, BPG deaminase and ferrohalatase, are actually regulatory points in erythroblasts. And also the biosynthesis of heme in erythroblasts depends significantly on the iron bioavailability for these cells. We're going to move on to the second molecule, and that is glutathione, GSH, gamma-glutamyl, cysteinyl, glycine, so on, tripeptide, which structurally represented here in a figure. So we know a lot now about glutathione. We know that one of the major roles of glutathione is its antioxidant function, but glutathione is also involved in numerous detoxifications, and you're going to discuss about it later on in some upcoming chapters. It's used for transport and biotransformation of numerous endogenous and exogenous substances. But the major point of action is the ability of SH group, which are functional centers of glutathione, so to be oxidized in disulfide bond. So reduced glutathione with sulfhydryl groups is oxidized to disulfide, oxidized form of glutathione, which does not have the physiological function. When performing its biological function, reduced glutathione is oxidized, and this is achieved by the action of enzyme glutathione peroxidase. We have already mentioned the role of glutathione peroxidase in the removal of hydrogen peroxide and the removal of reactive oxygen species. And we have also mentioned that reduced glutathione can be regenerated from its oxidized form by the action of enzyme glutathione reductase. So this is something that we have already discussed extensively in our previous chapters. Glutathione is also used for the biosynthesis of leukotrienes and in general for the maintenance of SH group metabolism. So glutathione is synthesized throughout two reactions catalyzed by two enzymes. The first one is condensation of glutamate with cysteine, catalyzed by gamma-glutamyl cysteine synthetase, and one molecule of ATP is required for the formation of new chemical bond, of the new peptide bond. So we have gamma-glutamyl cysteine formed as an intermediate, which is subjected to the reaction number 2, catalyzed by glutathione synthase or synthetase, involving the third amino acid, glycine, and for the formation of the new peptide bond, one more ATP molecule is involved to form glutathione, gamma-glutamyl, cysteinyl, glycine. Degradation of glutathione is slightly more complex, and it involves the reaction of 4 enzymes. So the first one is gamma-glutamyl transpeptidase, which actually degrades glutathione molecule in a way that one amino acid which is located out of the cell is going to be attached to gamma-glutamyl moiety of glutathione, and cysteinyl glycine is going to be removed. Amino acid which is attached to gamma-glutamyl group and moiety now passes through the membrane and enters the cell cytosol. Now this condensate actually is subjected to the second reaction catalyzed by gamma-glutamyl cyclotransferase, where amino acid which was imported into the cell is now released in a cell, and gamma-glutamyl is converted to 5-oxoproline, so cyclization of the molecule occurs. And finally, oxoprolin is hydrolyzed by 5-oxoprolinase and with the involvement of one molecule of ATP to form glutamate and ADP and inorganic phosphate. And finally, the cysteine glycine formed in the reaction number 1 is degraded by intracellular protease to cysteine and glycine. So each of those 3 amino acids is now released and able to undergo the biosynthesis of the new molecule of glutathione. Let's move on to creatine. So we all know that creatine is one of the very good and fine solutions for muscle cells and one of the good ways to store high energy bond instead of storing them in the form of ATP, which is not very convenient. So those high energy bonds are actually stored in the form of creatine phosphate, and when energy is needed in muscle cells, creatine phosphate hydrolysis yields to the formation of ATP, which is used for muscle contraction. So the biosynthesis of creatine starts in the kidney and it's completed in the liver. So in kidney, as you can see from the figure, glycine undergo reaction with arginine to form guanidinoacetate. Here, guanidino group from arginine, which was actually previously used for release of urea in urea cycle, if we remember. So now, guanidino group is transferred from arginine to glycine, forming guanidinoacetate, and the rest of the arginine molecule forms ornithine. Guanidinoacetate is then transported to the liver, and in liver it undergoes methylation reaction, and the donor of methyl group is S-adenosylmethionine converted to S-adenosyl-homocysteine, and creatine is formed. Creatine is secreted to blood circulation and transported to brain, heart, or skeletal muscles. In tissues, creatine now captures phosphate group from ATP to form creatine phosphate, and the energy is conserved in this molecule and high energy phosphate bond, and the reaction is catalyzed by creatine kinase or creatine phosphokinase. Creatine phosphate is unstable molecule, so it undergoes spontaneous cyclization, non-enzymatic reaction to form creatinine. Creatinine is not a functional molecule, so it's going to be excreted in urine. Obviously, the quantity of synthesized creatinine depends directly on the muscle mass. And because of its unique excretion pathway by kidneys, it's used as one of the tools for estimation of kidney function. But more about the role of creatinine during our practical exercises. And finally, we have serotonin, which is chemically 5-hydroxytryptamine, and this is the molecule synthesized in all cells. Actually, the highest amounts of serotonin are found in intestinal mucosa cells, and less quantities are found in thrombocytes and central nervous system. Serotonin is attributed numerous physiological functions, and some of those are perception of pain, regulation of sleep, body temperature and blood pressure. It's responsible for our mood control and appetite regulation, in the terms that low serotonin levels caused an increase in appetite and depression. So the third generation of antidepressants are called SSRI, selective serotonin reuptake inhibitors, because they're actually based on prevention of reuptaking and removal of serotonin from circulation, which prevents the formation of depressive states and moods. Serotonin is synthesized from amino acid tryptophan, and here is the pathway of biosynthesis. So tryptophan is hydroxylated, as you can see. So the benzene ring of tryptophan is hydroxylated by tryptophan hydroxylase. And as you already know, for hydroxylation of ring structure, we require BH4 as coenzyme. In the upcoming second reaction, 5-hydroxytryptophan is decarboxylated by DOPA decarboxylase, requiring pyridoxal phosphate as a coenzyme, so serotonin is synthesized. Further on, serotonin can be converted to melatonin in pineal glands in response of the dark light cycle, and the concentration of melatonin is increased in dark. So serotonin is subjected to reaction with acetyl coenzyme A, so N-acetyl serotonin is formed, and additional methylation of OH group occurs by S-adenosylmethionine, and melatonin is synthesized. Serotonin is catabolized by monoamino oxidase A to form 5-hydroxyindole acetaldehyde and finally 5-hydroxyindole acetic acid, which is excreted from the body. So these are some of the examples of important biologically active nitrogen compounds which are synthesized from amino acids, but this is not all. We have already mentioned histamine, which is synthesized from histidine by decarboxylation. So histidine decarboxylase removes carboxyl group from histidine to form histamine, which is a very potent vasodilator and which is actually a molecule which is released during inflammatory and response or allergy reactions. Also, a number of hormones, catecholamines, are synthesized from amino acid tyrosine, like DOPA, like dopamine, norepinephrine, epinephrine. From tyrosine, we have the synthesis of thyroid hormones and many others. But those specific pathways and consequently important physiological function of these molecules are going to be explained in more details in some other chapters. But until then, I can say that this was all that was actually scheduled to say about protein amino acid metabolism. So the upcoming videos and upcoming lectures will be something totally different, totally new. Probably we're going to move on to nucleotide metabolism. But until then, please stay healthy, stay safe, and bye-bye.